forgot to stay and lecture until the end, which just doesn't pertain to most of you, because I'm preaching the fire, because you're here, because you did the pre-lab. But I was telling one of your classmates, you know, when you don't do the pre-lab, you can't come to lab. And that's not necessarily like a punishment. Sometimes, like this particular classmate, it wasn't their fault. They were like they had some issue where they had no access to WBSU online. So because of that, they couldn't do the pre-lab. But because they didn't do the pre-lab, that means they're not prepared to do the lab. Therefore, they can't come to lab. I told them I'm going to meet with them and I'm going to go through the lab with them one-on-one. So it's not necessarily a punishment, but if you didn't do the pre-lab, you can't come to lab. So again, as I go through attendance, if I say you can't be here, don't be offended. But I've already left comments on it. Maybe they're hard to see, but you got to check your email. So when I leave a comment on your lab, your free lab, it'll say you should get an email that says, hey, and I'll ask you for something like, hey, don't forget to do this. You have to do this before you can come to lab. So you should be good. You and I already talked. But anyway, and you're okay. And also, I know some of these people aren't going to be here, but call them the name anyway, just because it's recording. Well, actually, is it recording? Yeah, we're recording. Yeah, we're recording. So this is more like just more stuff. All right, so Bangura, not here yet. Case Bolt is here. Tristan's here. Covington Griggs. No Covington Griggs yet. We still have 2 minutes. Devere. Dow, I thought, yep, Dow was here. Hughes. No Hughes. Okay. Lebdy is here. Carla's here. Dankins is not going to be here. Spoke to him. Mathis is here. Rains is here. Reed is here. Elise. Okay. Thompson is here. And Yin Dewey. Right here. Very good. Elkins up right there. Yep. And this is Elise, correct?
Yeah.
Elise is here. All right. Some people, for some of you, I know your first name, and others I know your last name. There we go. Elise. It's 10 o'clock. All right. To give people some more time before I lock the doors. So as a heads up for next week, let's talk about this really quickly so the same mistakes aren't made. So once again, after you turn in your pre-lab, which is due on Friday, after you share it with me on Google Docs, you're not necessarily done, right? I will leave a comment if there's something missing. So you have 2 things you should do. Check your email because you should be getting, like when I leave a comment, you should get an email and say, hey, you left a comment. Or, go back and check, right? Check to make sure you're good. Because like I was telling Tristan, it's a good point. Like there's no way 125 students. So if I say I need something from you, leave a comment. I can't just write myself a note and just keep going back, constantly going back for my students and make sure they turned in everything that I asked. You know what I mean? I know it sounds picky, but I just can't. Logistically, I can't do it. And it's also very important that you're prepared for that. This is a class, a team effort. So when you're not prepared, you're dragging everyone else down. So that being said, here's what's due next week, or excuse me, this is the pre-lab for next week, right? I want to show you really quickly, really big letters. Make sure you watch that video and answer those questions. You guys good there? Okay. Then after you read the introduction, you're just going to answer a few questions, right? As usual. And then in big green letters, you're going to keep going. Once again, just like this week, you have 4 sets of hypotheses. These are a little bit, I won't say harder or easier because there's fewer of them, but they're not multiple choice. So leave your hypothesis for that question, for that one, right? Then you scroll down to this set of hypotheses, or just, right there. Then you scroll down to one more set of hypotheses. And then finally, you scroll down to one more set of hypotheses. Then there's also a vote. You don't have to do this, but there's also one more thing that sometimes I do and sometimes I don't do. So it'd be nice if you were to take a look at it and tell me what you want to do. Should we do that? You can read it yourself, but that last one, I want your vote. Do you think we should do that experiment or not? Because it's already 4 experiments, so I'll let you guys decide. So any questions about that? All right, that's for next week. Let's talk about this week. The beauty of this is, again, this is why one of the reasons I'm so picky about the pre-labs is I know you've done the pre-labs if you're in here, so I don't have to spend a lot of time talking about this pre-lab introduction. We can just pretty much jump to the pre-lab questions. And then we can jump to the experiments. And carbohydrates. So this is not really an important question. This just proves you read it. What's the ratio of carbon to hydrogen and oxygen in carbohydrates? Does anybody know what the ratio is? Very good, 1 to 2 to 1. And again, to reiterate, that's not really important. You're not going to need to know that for this. You're not going to need to know that for the exam. It's just proof that you read the introduction. Anybody give me an example of a polysaccharide? I think so. Starch is good. Specifically, starch is the polysaccharide. Yeah. So those actually will be important for the exam, like starch. You'll need to know starch. What else? Chitin, glycogen, cellulose, stuff like that. Here, this one is important for understanding today's laboratory experiment. Can you use an unknown substance as a control? That's the first part of the question. Yes or no?
No.
No. Good. Very good. Why not?
That's unknown.
Yeah, it's unknown. You don't know what's in it. It can't be a control if you don't know what's in it. That'll make a little bit more sense here in a second, just in case that's not making sense yet. Really quickly, how would you say, and this is also slightly important for today's experiment, the difference between qualitative and quantitative analysis. Anybody want to give that a stab?
One is like numbers, and the other one's like information.
Yeah, right. So quantitative, like did you actually have data like numbers, like you said? Like if you were to measure something like, hey, which fertilizer is best for these plants? You measure those plants and take those numbers every week. And today what we're going to do is qualitative. So for example, kind of see it here. In this first experiment, you're going to see the level of monosaccharides in these substances, right? We're not going to have a number. We're not going to say how many parts per million. We're just going to be like, all right, there's none, or there's a little bit, or there's a medium amount, or there's a lot, right? So we're not actually using numbers. This is qualitative. This one's really important. You need to know this for today's experiment, not for the exam. The difference between a positive control and a negative control. Anybody want to take a stab at that one?
Is positive control, there's nothing more in the experiment than the positive control?
That's true, yeah, that is a true statement. Most people answer it that way. Not what I was going for, but it's still correct and still in the introduction, so yes, that is correct. What I'm getting at with positive control is like when we do and I'll give you an example here in a second, when you do an experiment with different substances, if you know the thing you're testing for is in there, that's a positive control. If you know it doesn't have whatever substance you're testing for, that's a negative control. Does that make sense? All right, so really quickly, let me give you some example of that. Let's say, turn the slide back on now. Let's say I came up with this new invention where I could tell you, if there is alcohol in a drink or not. Right? Maybe there's some new thing going around where bartenders are pretending like they're giving you alcohol, but they're not. You're pissed off about it, so you just you come up with this stuff, and you put it in a drink, right? You get a drink from a bartender, and before you go out to the bar, you test it. So first you put it in the thing of, oh man, I don't have the right colors. Anyway, let's say you put it into something with pure alcohol, right? You got these drops, just to test it. You want to see what it does before you go to the bar. You put it in there, it's pure alcohol. Let's see, let me think about this. Let's call it, I don't know, green. Right, so it turns green if there's pure alcohol in it. Then you put it in pure water and it turns yellow. Right, as you do this before you go to the bar. So now you want to see, is this bartender's giving me weak drinks? So they give me drinks with no alcohol, whatever the case may be. You go to the bar, you order a drink. Hold on. Right, this. Again, this is from your little calibration before you went out to the bar. This is at home. Water, pure alcohol. Right, water's yellow, pure alcohol's green. All right. So you go to the bar, put it in the cup, and it turns yellow. Is there any alcohol in there? If so, how much?
Yes.
None, right? If it's pure yellow, it's none, right? Because you know from earlier, when you put it in water, it went yellow. You put it in, you get another drink, you put it in there, it turns it's completely green. Is there any alcohol in there? Yeah, there's a lot. You put it in and get a third drink before you get cut off, and it's yellowish green. How much alcohol is in there? Right in the middle, right? That's what you're doing here, right? So you're having all these different experiments, and we're going to help you. You guys are going to break up in teams, and you're going to have a positive control and a negative control. And for these tables, negative control is always listed first, positive control is always listed second. And that's all you're going to do, right? So, for example, with this first experiment, there we go. You're going to have, we're going to need more test tubes too, because we need, let's see, 9 times 3, so 27 at least, maybe 30 just in case something goes wrong total. Anyway, so for this first experiment, you're going to have one that has pure water in it. You're going to have one that has pure glucose in it. You're going to mix it with a reagent, which is just a certain chemical, like the example I was giving you right there. Then you're going to do some other stuff for the water bath. We'll talk about that later. Then you compare them. And for the rest of these, like you're going to look at the starch one. Say, does it look more like the tube of water or the tube of glucose? If it looks more like the tube of water, you say it has less monosaccharides. If it looks more like the tube of glucose, you say it has more monosaccharides. Does that make sense? I hope it does. And that's it, right? Same thing here. Second, other experiment. You're going to have a tube of water and a tube of starch. And you're going to compare the rest of those tubes to those two tubes. Does it look more like the negative control or the positive control? The lipid ones, that's a little bit different. You're going to have some paper. Yep. You're going to put some water on there, which is still your negative control, just to see how water looks like on a piece of paper. You're going to put some oil on there because that's your positive control. You can see what that looks like on a piece of paper. And then again, for everything else, you're going to compare it. You're going to rub a chip on there. Does it look more like the water or the oil? You're going to rub some bread on there. Does it look more like the water or the oil? Does that make sense? And then finally, again, just like these other two, when you're doing proteins, you'll have a tube of water, you'll have a tube of pepsin, which is a type of protein, and you're going to compare them, right? So you're going to compare the starch tube. Does it look more like the water or the pepsin? Does the sucrose look more like the water or the pepsin? Does that make sense? So that's the big picture. When we're doing this, there's a lot of, I'm sure you've read it already, there's a lot of little steps we have to take. I just don't want you to get lost in the steps and miss the big picture. So that's the big picture. We're comparing all these other ones to the negative and the positive control. And since it seems to be such a rough lab, just to make things a little bit easier for you, when you're looking at these tables, for the first table, I've declared those extra credit. You can put them in there if you want. The colors, write it, don't write it, I don't care. But if you do it, it's extra credit. So if you want to save time and make things easier on you, don't do it. If you want to get some extra credit, do it. Same here. For the second experiment, it also asks for the colors. Put it on there if you want to. Don't put it on there if you don't want to. Good? And I'm going to come up with some other extra credit to help you guys, make up for any lost points. So we have to wait for him to get back with these tubes so we can finish this. In the meantime, I'm going to break it. Well, let me explain one more thing. Sorry. So you guys are going to be broken up into teams. Your team is just going to do one experiment, right? So there's going to be 4 teams, each one doing their own experiment. You're going to come up and you're going to write your data on the board. So all of you have to do the whole lab worksheet, but you're only doing the one experiment. Does that make sense? So feel free, for example, and you will be team 1. She's going to do team 1, right? So feel free when you're done and you're doing the lab to talk to somebody who was in team 2 or team 3 or team 4 if you're like, I don't quite understand what your results mean. So you guys talk amongst yourselves. You're here for each other. And that's it, I think. I think we can get started now. So yes, your team 1. Tristan, your team 2. Team 3. Team 4. Team 1. Team 2. Team 3. Team 4. Team 1. Team 2. Team 3. Team 4. Team 1. Right? All right. So once he gets back with all the stuff, we'll get started. And you guys have your computers, right? So you know where the directions are. If you click on the link, it'll show you the directions, and he and I will be here to help you. So, team 1, you should go ahead and get started first. I'm going to help team 1 first because theirs takes longer. So, those are your positive and negative controls. So then you'll have 7 other. Anyway, team 1 should meet here, team 2 should meet there, team 3 should meet there, team 4 should meet somewhere in the back. Sorry, I didn't say that. 2, 3, 4. Thank you. 5, 6, 7. All right, so there's that. All this stuff. Team 2, here you go. Again, you're gonna have your first 2 are your positive and negative controls. You guys are gonna need to grab a marker, at least one marker for each team, because you need to write on these tubes so you know which is which. And the markers can be found up front on that card. 1, 2, 5, 6, 7. Team 3, I'm helping you last because you have the easiest of the experiments. Yes, 2 and 7.
Anything one, two?
Just want to make sure they're very aware of their. Sorry, we're doing the second turn? Yeah. Okay. So you have any markers you can like write.
No, but there's stuff.
Appreciate it. And I'm gonna grab some more stuff for you guys. But of course, we don't have to wait for you to grab it.
What's on the check?
I'm trying to-
It's how
The
much?
plus, that's what we're actually doing. So like if there's a lot, then we put 2.
Oh, so we're just on that.
Yeah.
Thank you for your help.
Well, we have to wait for them to get it.
You guys already have markers, right?
Yeah.
Okay.
Jesus, click on this link and open it up.
We need more people on team 3. So somebody didn't, I guess maybe pointed wrong or something, but let's see. You guys are 1, 2, 3, 4 for team 1. No, wait, 3 for team 1, 4 for team 2. All right, can you maybe come to team 3? You don't mind? Yeah, we'll have 2, 1, 2, 3, 4, and then you're team 2, correct? Oh, you're team 1, okay. Yeah, if you go there, that'll be fine. 2 will be just fine. You guys have the easiest experiment. Here's what you need. Yeah, detecting liquids, sorry. I don't remember if you specifically need that or not, but just in case, there you go. Basically, what you're gonna do is put in the test substances, and you're gonna come up here to do it.
What is the 1 centimeter long?
You'll have to use the ruler.
Yeah. Oh, yeah, I'll just give it to you right there, yeah. Good question. So, yeah, you're gonna put in the test liquids.
Yeah. Make sure you.
Good job, I'm glad you read. So, yeah, you're doing the Benedict's reagent. So what you'll do, like you said, the 1 centimeter mark, right? And you'll put each test substance in, and then you put this stuff in. This is all written down for you, but I want to verbalize it. And then once you filled it all in, you know, put it in that hot water bath over there.
Okay.
Okay. Yeah, so you have your marker, right? And make sure you mark
Yeah,
which one's which, so you don't like lose track of which test substance you're dealing with. All right, and I don't remember if you guys need a marker or not, so. But if you, all right, you need one. All right. Yeah, don't worry about that. Yeah, as long as you know,
So right now, we just need more complicated.
you just have like
1 centimeter.
one of these for each thing, right? So there's your water,
So one small.
there's your oil, there's bread, oil chip, all of them? Yeah. Bread,
The same color too?
cookie, what, I heard to write the name on it, don't worry about it.
Okay.
I
Oh,
just wanna, like I
that's the
said,
line.
compare, you know, put a little bit of water on here, put your oil on here, wait a little bit, and just look at it, and then compare, you're gonna compare the rest of them to like this water, control, which is vegetable.
Once we get them all,
It's more like your negative control, you see it has no lipids, no lipids.
How many?
I've been going to get some markings on the drum.
So the negative control has water, right? So negative control, yeah, you're gonna put some water on there.
Engineer or
You
something?
might want to write H2O or water or something, just so you know. Because
What's
you
that?
know for a fact that water doesn't have oil in it, right? It doesn't have any lipids, it's pure water. And then here, you're gonna have oil, and that's pure lipids. Some water on here, some oil on here, and that's gonna be your point of comparison. That makes sense? So then, like, for example, on this one,
Everybody else.
you're gonna rub some chips on there, chips, right? And you're gonna compare it, it's more like the water, the oil. It's more like the water, you see it has, doesn't have a lot of lipids. or none.
Which ones have you already marked?
It's more like the oil.
Which ones have you already marked?
Did I give you guys this what you need? This is your iodine, should say iodine, but it is iodine. You'll need that, and yeah, maybe all the other test substances you're gonna need are up there.
Did you do 1 and 3 or just 1? You need to do 1 and 3. Yeah.
You said two?
Yes.
So I got something
Didn't he say we
Okay,
could?
then I have
that, however long,
Okay.
whatever.
I don't think
Test substance.
it just brings it back
So
every
the
year.
rest of this.
But they're all different. Yeah, he said we gotta do it up there.
The water's right here, the distilled water.
Oh, I wanna stand up.
We can put these in the test tubes.
Yeah, hey, you don't need
No,
this too.
they need to put it on the front.
Yeah, you're gonna need these, I'll get it for you.
So we're gonna have to label all of it. We used to do these papers in like 3 weeks. I like how it's usually starts to look
Chips, sorry. Red chips, but we don't have chips, huh? We don't have any chips?
I'm done with
We
the.
don't have any chips.
Yeah, Dr. Sutton.
Okay, yep, you guys skip that part, I guess, or unless you answer. Okay.
I don't want us to mess up the measurements.
Oh, perfect, okay, thank you. He's on it.
So where do we get the, like, Epsom starch?
Everything will be up here unless someone's moving.
Oh, okay, okay.
So let's see,
And then. So I can bring the tubes up here, fill it up here?
yeah, or you can bring them back there as long as you bring them back, whatever works for you.
Are y'all ready for me to get the water?
So we go to the shit. We just squish it there.
And then the nice one is a question mark, what are we doing with that?
Who's got the water?
This one?
The nice.
Oh, sorry, yeah. So the question mark, if you look at the table, it says unknown solution. I was just being lazy, so instead of writing unknown solution, I put a question mark. But this is what you'll use for the unknown solution. Your guess is what it is? That was my guess too. And normally I would not recommend doing this. Don't ever just smell things randomly in the lab.
Oh,
But I happen to know what it is, I think, so I'm gonna smell it. And actually,
it's milk.
that's what I think. I think it's, I mean, I don't know for sure. I took the risk by smelling it, but I'm pretty sure it's milk with a pink food coloring. And I think they do that to throw you off, because it does look like that. Not that it matters. You don't have to figure out what the unknown is.
It was milk.
You just,
Yeah.
you just have to do it. Oh, really? All right.
Yeah, the Benedict solution. Read it and just.
So just so you guys know, yeah, what you'll do, not just the water, but you have the test substance for every single one, like one with water, one with glucose, one with fruit juice, so on and so forth. And then, once you have them all filled with the test substances, then you put that blue stuff.
Lucas 3 starts to.
And the example I was using about taking the thing to the bar, that blue stuff is your magic chemical to see. It should say it on there. Yep. So there's been an update. We don't have chips. So you guys, I'm only telling you that way when you're doing the lab and you're like, why is that blank? It's blank for a reason. I'm gonna write NA. You can write NA as well. And if for some reason I get stupid and mark that wrong when I'm grading your lab, remind me, say, hey, stupid, we didn't have chips. I do forget things, especially this semester. It's been a rough.
You got the water?
Oh, yeah. Get notifications.
I'm gonna,
Puts a comment on here. I've never got one.
So, well, from which
How much will go down right there?
Well, I think when he turns it back to you.
Okay, so, what are you doing? I mean,
When I,
yeah. So you
When
fill
he
that
turns
first line
the
of water,
page back to me, or like gives me the grade,
fill it all the way up.
that's where I see the movements. So it's not like,
then with this one, you fill the
Like
first
you'll
line up
see
with glucose, and another two.
That it
Yeah, that's what I'm
graded it.
doing.
yeah.
Oh, I'm sorry, yeah, so yeah,
Say
same,
like-
same, same
So
with the
you
rest
back
of
your
them.
grade.
You
and
fill
it'll have moments.
First line up with Pepsi, and then you use that boost up.
Has like one.
The prescription is gonna come
Sucrose, huh? Keep in mind, you guys are testing monosaccharides. Not that you're not gonna lose points for that.
Juice? I think it's also. I think it's the other.
It doesn't say it, it looks like lemonade.
I'll have it.
Have you sold it yet?
Oh, yeah.
Okay, that will be number C.
Are you doing the water one first?
I'm ready.
Here's water.
Glucose? Okay, well, they're all numbered, so we'll have
Okay,
to.
glucose,
Yeah.
I don't know where to put it at, that's why.
glucose.
Should we do
Say
color? Yeah, we'll this one.
Pepsi, what's Pepsi? 7. And that's the
That
water.
was that.
Yeah.
What? Oh, this one.
Then I'm gonna get us glucose.
Here it is, glucose.
Okay.
Where do we put these?
What do you need? Oh, when you're done with them? So the little things, you can just keep them in their respective areas, or you can just put them on this table and we'll take care of it. Or cart, I should say.
We do glucose here.
Yeah, we just need 8 and 9. We need the oil
Yeah.
and then 9 is or what was already.
You just took 9 is a question mark.
the pink stuff.
You guys go with the glucose?
You got the big one?
Glucose.
You did glucose?
Grab this quick.
You took a pen today.
Is that the juice? Here's the fruit juice.
Write some hints on the board to make things even easier for you guys. For when you're doing the lab. All right, so we didn't have any markers, right? You used dry erase markers?
Yeah, I had to do it.
So just be careful then, since you're putting in a hot water bath. Make sure you guys know which is which, because the dry erase might wash off in the hot water bath.
So you recommend doing it one at a time instead of all together?
Yeah, that would take you forever. I mean, that would be better, but it would be unpractical for you. So just make sure you know what order
I
they're
try
in.
to write them big. Hopefully they stay.
Yeah, and you put them at the top. It's very good.
Here's our eggs.
Very good, guys.
So gently mix it and then we'll all, and then we'll put them all in at once. That way, after the 5 minutes, we'll all right.
So if you ever get confused about your results and say you're not really sure, that is legit. You can put that. So on some of those spots on the table, if you really can't tell the difference, you can put inconclusive. I mean, obviously, it'd be a little suspect if you did that for every single one of them.
So-
But if there's one of them and you just can't really make a choice, you're allowed to put inconclusive. Yeah, you put them all in
So
together.
you do the first one, good question, and then you'll use the.
And that'd be mostly for you guys too. With yours,
First step goes to the second.
that'd be A's, you know, things.
8.
So if there's any
Oh,
other
means
that
marks,
can't
too.
tell for sure, you're allowed to say inconclusive or not.
Oil 8, right?
Allowed to put that on the table.
Yeah.
And then
What numbers we need?
We didn't do water either.
We got water. Oh, no, we didn't.
We need Pepsi, right? There's 7.
So give some hints here.
We need number 4, what is that?
Four sucrose.
Anybody got that estimate?
Starch and water.
One and two.
That's my daughter.
We don't have one, and I'm-
Sucrose is 4.
Yeah, okay.
Where's that water? Is this one water? Sure. Oh, wait, one.
I have oil. We know the starches.
Yeah, I don't have
I've already done 3 today. Yeah, 3's are. So we have 1, 2, 3, and 5 already done.
Place and drop.
She's doing 4, you're doing 7.
We need 8 and 9, right?
We still need to sit down thinking about the egg roll cabinet.
Okay, I'm number 8, I'm doing 4 off right.
So 2 to 3 drops in water and 2 to 3 drops in starch.
I'm glad y'all did them, or I'd have been crying.
Yeah. You know, I think that's what it is.
And then that one you just start.
And 2 to 3 and all the other ones.
Because if you don't start the ingredient,
Then the ones I did, I'm sorry. It's why I did it too.
is that number 8?
Yes.
So the vegetable one, oh yeah, so you can see the vegetable one.
Yeah.
It separates, isn't it because of the density?
It could be the density, it could be, the chemical, like one's hydrophobic, one's hydrophobic.
I just think of all the words.
So a heads up for you guys to make your life a little bit easier. After every table, you have 2 sets of questions. The first one, you're gonna describe your results, like there was this much, there was that much. Only do that for the test tubes, right? You don't have to talk about your positive control, your negative control when you're talking about your results. Because you weren't testing your positive control and your negative control. That's what you're using to compare your test substances. So to make things easier for you, make sure, you don't write about your positive or your negative control.
You tell me.
How was only doing the.
Yes, yeah, because just like I was saying,
Everybody
everybody
has their.
bars here. Like that's your magic liquid to see,
At the bottom.
how much protein is in, right? So you gotta put it in the protein, that's what it looks like.
We get the data.
Put it in the water, like, that's what it looks like when it's pure water. And then you compare the rest of them, like, does it look more like this is pure protein than the pure water? Glad you asked. Color? Bluish green?
So this is starch, right?
Yeah.
We did number 7, right? All of them done.
Show for no starch, I guess the dark.
Now for you guys, probably, even though it says it on the table, more importantly is what it looks like afterwards, right? Because you want to be able to, compare your test substances to your positive control, your negative control.
This lab is.
Yeah, well, if it's if there's none,
Y'all want me to write the color on so we can
oh
get
yeah,
it?
if there's none, you can write 0.
This is the water.
There was a little bit, right, +1. There was a minimum amount.
Yes, that's just the water.
That's fine.
So the darker the color, the more starch it has.
Matter of fact,
So these might have a little bit.
5 is green, right? It's
Yeah.
green, right?
Yeah. These have no
there you go.
starch.
Seems like you guys
3 and
understood.
4, no
Oh,
starch.
All right, awesome.
Is there any that have a little starch?
Seems like not only did you get it done, you understood what was happening. Very good. So on these tables, I'm going to go ahead and write this in for you. The negative control obviously is always going to be at 0. Because we know it has 0 in the substance. The positive control is always going to be 3 pluses. We know it has, it's pure, whatever it is that you tested, pure glucose in this case, pure polysaccharides in this
You don't
case,
tell me the water and stuff, you know, I mean,
pure protein in
It's on
this
my
case.
Clear lows, a little moderate.
So what are we talking about?
So like, the colors, so the more it looks like that, the more starch it has. So then we're gonna record those 0,
Wait,
1
wait,
plus,
wait,
2 plus, or all less, the more.
7, 8, 9.
So water, it's all my.
That's a lot.
2. And I feel like this is.
Well, we don't do these. No. These are our,
So water.
these are our controls. We're only doing filters.
Water up plus.
No, we don't do water or such. Those are the controls.
Good. So then this would
you
be
want
like
to bring
These
the
are a lot. This is probably moderate, and these are little moderate. These are nothing. So.
nothing.
Yeah, these are the same as water. So we do, what we do to some of this, what we do, just copy that of it, the colors.
Color before heating for all is yellow.
Yeah. And then now polysaccharides, 0, plus. Or starches.
Starches.
Rounds.
Say dark brown, and then everything else is yellow.
And then glucose is only 3. So 3 and 4 are 0.
And 4.
So as I have written on the board, you will need one graph, and it's going to be the graph of the experiment that you did,
And the other ones are like.
not all of them, just the ones you did.
Is a little.
Anybody want to guess what kind of graph it would be? Between a line graph or a bar graph? Think about the data you're taking here.
It's also
Bar.
plus.
Is it distinct data or is it continuous data?
Right.
Distinct, right? It's either this much, this thing has this much, this thing has this much, so bar graphs, right? But for extra credit, you could do graphs for all of them if you want.
7. So 6. It's like plus.
So I did record this, the data's up here.
Is burned.
I'll be posting that video to the
7.
It's like orange.
YouTube playlist. So while you're doing the lab on yourself, if you're working on it, if you need that video, you can find it on the YouTube playlist. If you don't know where to find that, shoot me an email and I'll send you a link. That way you can be reminded of all the stuff you guys did.
We'll make the bar graph
So
on.
8.
Okay, make it however you want. You can do it on a piece of paper, you can do it using a computer, however you want to do it.
What did he say to put undistinguished?
Yeah.
Now,
He said that was.
So it's sensitive to heat.
Is that? No, yeah, you write it there because everybody else is gonna need your data too. Likewise on your computer, even though you're not
Well,
doing
just
these
copy.
experiments. Yeah, exactly. So good, so you guys are timing, so just to be clear, while you're waiting on it, just so you guys know, so thank you, Jude. But you're gonna have that tube that has water in it, you're gonna have a tube that has glucose in it, right? And also, they all have the magic potion. And then you're gonna compare every single one of these to those two, to the starch. You can say, does it look more like the water or the glucose? If it looks like the water, you can say it has no saccharides or few. If it looks more like the glucose, you say it has lots of saccharides. Does it make sense?
There's 3 old basically.
Yeah, exactly. It's again, like I was saying, using the bar example, you got, had your little magic alcohol potion, alcohol detector. It'll look more like a glass of water or a glass of pure alcohol. So compare each one, right? Compare this to this, compare this to this, compare this to
everything is
this.
compared to the water.
Yeah, everything's compared to the water and the glucose, right? Just want to see, does it look more like the positive control or the negative? That goes for everybody, that's how it's gonna work. And you guys don't forget to put your data on the table, so for your classmates, because your classmates will need your data. Team three's already done, like I said, they have the easiest. And of course,
Yeah, so like, do 1, 2, and
muddy the
3
water,
least
good
of
at that.
So you're doing 1 for each of the twos. But there's none.